All right, welcome everybody. We're back here for class 3, and we are going to be talking today a little bit more about toxicity that we started talking about in our last class. If you don't mind, like usual, can someone just give me a quick thumbs up in the chat if everything looks and sounds okay? And if it does, I can continue, and if not, I can make some adjustments on my side here. I think everything looks all right from what I can see at the end. If the sound seems a little off, too high, too low, just let me know in the chat and I can adjust it on the fly. All right, so if you remember what we were talking about last class, we were talking mostly about toxicity in the acute sense. And what I mean by the acute sense is toxicity that all right, thank you for the chat. I do get like a 15-second delay, so it's it's nice to see your responses at least a little bit after I ask something. So we were talking about acute toxicity, which means toxicity that'll cause you immediate harm. And in the context of something like alcohol or drugs or poisons or things like that, what we're really talking about is poisoning yourself acutely. It means sort of to overdose, to have enough of this compound that it causes your body to shut down or die or some other negative effect. And this was to be distinguished from what we call chronic toxicity, which we're also going to talk about in some detail today. So thanks everybody for the thumbs up there. So we identified last class a measure of the acute toxicity of different chemicals that we called LD50. And the LD50 was the dose required to kill 50% of some test population. So we did look at that, and we were able to categorize compounds, and this is, I think, right where we ended last class. We were able to categorize different chemicals into the following groups: super toxic, extremely toxic, very toxic, moderately toxic, slightly toxic, and practically non-toxic. And these characterizations are based really on how big of a dose is required to kill 50% of some population of adults. We did talk about how these numbers are often measured on animals and then extrapolated to human beings. However, in some cases, in the case of especially things that humans maybe more frequently ingest, like drugs and alcohol, we do have these data for actual adult human beings as well. So the LD50 measured here on mice. And so the first thing I wanted to kind of touch on today is this idea that human beings are not people. Is this animal testing and then extrapolating to human beings something that's valid, like something that always makes sense and something that, you know, we can be confident in making that assumption? And the answer is yes, no, sort of, maybe. Okay? There are differences between different animal groups, and in particular, how a particular substance may impact one group of animals versus some other species of animals. And I'm going to start with the example today of chocolate. Now, chocolate contains cocoa. Cocoa comes from the cocoa plant. And chocolate contains naturally a chemical with the structure you see drawn here called theobromine. And a funny thing about the name of these structures, I don't know who gets to pick the names, but there is no bromine in the molecule, yet it's called theobromine. Maybe somebody early on thought it had bromine and it didn't, or I don't know what the story is behind it. Anyway, it can be found in chocolate, in cocoa, and this is a substance that is moderately toxic to human beings with an LD50 value of around 1,000 milligrams per kilogram. So if you go back to our table over here, you know, it fits into that moderately toxic category. However, the LD50 for the same substance in dogs is 300 milligrams per kilogram, which means it's a little bit more than 3 times more toxic. And this number, by the way, LD50, the lower the value, the more toxic a substance is, because it means the smaller dose that's going to be required to cause some negative effect with you, right? So what does this mean in a practical sense? This means if you weigh 70 kilograms, which quick math is like 180 pounds, at 1,000 milligrams per kilogram, you would have to take these Lindt Excellence bars. These are 100 grams per bar, so it's a big chocolate bar. And the higher the cocoa content, the more of this theobromine there is. So if we go for the 70% cocoa type, you might have seen this before, to hit your LD50, you would have to sit down and eat 83 of these chocolate bars in one sitting. That's a lot. That works out to what? 8.3 kilograms of chocolate. So maybe possible, but not a realistic worry for most people. If you did that, you still 50-50 chance of surviving it. For dogs though, dogs are smaller. Dogs, let's say you have a 20 kilogram dog, that's still a pretty big dog, right? Like that's a 50-pound dog, almost 50 pounds, 44 pounds exactly. What that dog would have to eat to hit its LD50 was 6 bars. And that's because the dog weighs less and the dog has a lower LD50. And so what that really means, you can imagine a dog, if the dog, 44 pound dog, big enough dog, if it was able to have free access to 6 of these chocolate bars, it could very easily wolf those down in one sitting. So this is why we often say that chocolate is poisonous to dogs, but not poisonous to people. The reality of the situation is both of them are toxic to both of us in certain quantities. The quantity for human beings is just so high at 83 chocolate bars that it's not a realistic worry, where for dogs it's small enough that it is a realistic worry, which is why we say keep chocolate away from dogs. In particular, if you went from 70% cocoa down to 50% or 20% or whatever, like Kit Kats are or whatever, the risk just goes down and down and down for your dog. So the risk is not linear. So you may ask the question, why is there such a difference between different types of animals? And the reason is we have a whole biology. We have an endocrine system, we have kidneys, we have liver, we have various enzymes and so on that we have evolved to handle the normal chemicals that we would come into contact with through our normal diets. And human beings have an extremely varied diet. So we would eat all sorts of plants and animals and really whatever we could find during our evolutionary period. Dogs, however, are carnivores, so dogs would not be, in their evolutionary years, exposed to the same range of chemicals that human beings would. So they would not have necessarily evolved ways to handle large amounts of theobromine, for example. So a dog might be more similar to a cat or some other carnivore in terms of toxicity of different chemicals, but it turns out human beings are generally better than carnivores at handling a variety of different chemicals that would be present in food. The heavier the organism, the lower the risk. That's right. And the reason is that LD50 is expressed in quantity per kilogram of body weight. So the more you weigh, the more of a certain compound it's going to take to kill you. Now, it's not quite that simple. It's not exactly linear with human beings, especially because, depending on your percent body fat, you could have different amounts of what we might call vasculature, and that could affect it. But roughly speaking, the more an organism weighs, the more compound it's going to take to cause harm in that animal. It turns out actually that for most toxic compounds, one of the best models of human toxicity is the rat. And the reason for that is that rats kind of have a diet like us. They'll eat anything. They'll eat meat, they'll eat vegetable matter, they'll eat almost anything they can get their hands on. So the types of diets that they would be exposed to in their natural world would be more similar to a human being's diet, especially rats that live in cities and actually eat human garbage, right? Rabbits are another maybe common one. Of course, rabbits don't eat meat, so it would be sort of the other side of the coin, more vegetarian diet in that case. All right, I want to tell you a story though about one such example of a compound, and this is maybe an outlier case where there was like an extremely big difference between how a compound impacted a human being versus impacted an animal. And it was it was a drug, but it wasn't ever approved. It was research was being undergone on this drug, and the name of it is there, Theralizumab, I think this is if you pronounced it, and it was developed by a professor at the University of Würzburg, and it's for treatment of, I think it's probably beta cell, chronic lymphocytic leukemia, and rheumatoid arthritis. And rheumatoid arthritis is an autoimmune disease, and what they were trying to do is get the immune system basically to stop attacking itself. And it turns out they created the drug. It actually worked fantastically well in animal models, and it caused an upregulation of T cells, which are immune cells. And what this had the effect on was to calm the immune system and prevent symptoms that are associated with overactive immune systems, which is many autoimmune immune diseases. So they first tried it on mice, worked fantastically well. They switched to monkeys, worked great. Then they said, okay, next step, we're going to try humans. We're going to do human trials here. And so what they did, they wanted to play it very safe. So they identified a number of patients. They took 6 volunteers who were suffering from, I don't know if it was leukemia or arthritis, but they started them out with what's called a subclinical dose. And a subclinical dose means it's a dose that's much lower than would be expected to cause a clinical effect or biological effect. Much lower than, for example, the doses that were given to the dogs or the mice and the monkeys. And the first dose they started with was 500 times lower than the dose that was found to be safe in animals. And the idea is, if the body tolerates that, you go up a bit, and you go up a bit, and you go up, I guess my hand you can't see, up a bit, up a bit, up a bit, up a bit. And you kind of increase the dose over time to see slowly and carefully if this is going to affect people. So they started the trial, and all 6 of them got this very low dose. Within an hour, all 6 of these men began to experience excruciating headaches, shivering, back pain, gut pain, diarrhea, swelling, and nausea. 4 hours, all of them had a fever, they were flushed, blood pressure was dropping dangerously low, hearts racing. They saw lymphocytes and monocytes, which are things you want, vanishing, disappearing very quickly. One patient, after 5 hours, was fighting for his breath. They're all experiencing lung pain. And they were given steroids, other drugs, to try to help them manage these symptoms. After 12 hours, the patient fighting for his breath was so bad that they were put in the ICU and put on a ventilator to keep them alive. The other ones were also taken into the ICU just as a precaution. One suffered a ballooned head. After 24 hours, so 1 day later, 2 of the people were on ventilators, and the 4 others were needing support with their breathing. After 48 hours, the 4 least affected began to recover, but all 6 then began to suffer multi-organ failure. They were attached to kidney machines for dialysis. One volunteer lost his fingers and toes. 5 out of 6 were eventually released from hospital within about a month. The last was in the hospital for 3.5 months. So this is quite a difference in the fact, a 500 times larger dose was very well handled by mice and monkeys. This is not a usual thing, by the way. This happened in this particular example because it was a compound which was known to have a direct impact on the immune system. So it was almost like it was a domino that started a chain reaction in the immune system that was causing all of these problems, as opposed to the drug having a direct toxic effect all on its own. Needless to say, though, while it's usually the case that we can extrapolate from animal experiments the impact of chemicals on human beings, every once in a while you get an example like this one that there's just almost no relationship at all, that we can't make that jump, which is why people really approach these sorts of studies with great care, why we give somebody a dose that's 500 times lower and observe them in a situation where we can get them to the ICU right away if need be. All right, we have another question here. One thing I want to mention about these questions is the first set of questions for Unit 1, which we finished last week. I've set the due date for that to be this Thursday, and I will just make double sure. I'll send one email just as a reminder, but you got to keep on top of these things as well. This one says, what chemical is found in chocolate and shows considerable toxicity to dogs and cats, but is of little concern to human beings? And we get arsenic, sulfamethoxazole, you might remember was the drug if you get a UTI, theobromine, and theralizumab. Theralizumab. Yeah, we'll go with that. The answer here is theobromine C. All right, apple seeds. So speaking of other acute toxins, there's a compound in apples, actually specifically in the seeds, called amygdalin. And amygdalin, again, it's a natural compound. It's produced naturally by the plant. It's found in apple seeds. It's found in the pits of fruits like peaches or kind of anything in that family, like peaches or plums, or what else is in that family? Apricots, things like that. It has this chemical structure, and what happens, the drug or the compound itself isn't that bad, except when it hits the stomach and our digestive enzymes begin to break it down, it releases cyanide. And actually, if you look at the molecule here, that CN part that you see hanging off the bottom of the molecule there, that breaks off and becomes CN minus. Come back. Becomes CN minus, which is cyanide, and cyanide is an acute toxin. So it's not that the compound itself is an acute toxin, but it becomes an acute toxin when we consume it, and it's found in the seeds in the apple. Of course, like any toxin, any acute toxin, the dose is what really determines if it's going to cause us trouble or not. So for this compound, for cyanide, the LD50 is about 1 milligram per kilogram body weight. Now, 1 milligram per kilogram body weight, that makes it about 1,000 times more toxic than the theobromine that's present in chocolate. Because that was 1,000 milligrams per kilogram body weight for human beings. So this means you need 1,000 times smaller dose to get that same deadly effect. So this categorizes it in the super toxic range, and that means to kill a 70 kilogram adult, you need about 70 milligrams of cyanide, which is not a lot. It's easy to get that dose if you're eating something with cyanide in it. To actually get this from eating apple seeds, you would need to eat about 183 apple seeds, which is roughly the number you would get from about 18 apples. So I don't know if you guys eat your apples whole with the core and seeds and all. I know people who have they eat an apple with the seeds. I guess animals do, like horses will eat an apple, they'll just eat the whole thing with the seeds and all. But the amount that it would take to kill a horse would be pretty large. So all I'm saying is that for whatever reason you like to eat apples, it's probably best to avoid the seeds, especially if you plan on eating lots of apples like this. Is that the cyanate ion that is toxic, or the compound it forms like potassium cyanide? So it's not the cyanate ion, it's the cyanide ion. Cyanate is something else. It doesn't form compounds in the body. The reason cyanide, I believe, is toxic is because it binds irreversibly to hemoglobin, kind of like carbon monoxide, because it's a similar shape and size to oxygen. But basically, I think I could be wrong about that, though. I should look that up, how it actually works. But no, it doesn't form compounds with cyanide in the body. It just stays a free ion all by itself. Do the apple seeds accumulate, or will the acid in our stomach corrode the seeds? Well, eventually you'll pass the seeds. If you ate an apple a day for 18 days, you're not going to have this buildup of cyanide that would be toxic to you. So yeah, your body, kind of any toxin that your body takes in, your body will find ways of detoxifying, excreting it, So this is why like small amounts aren't such a concern, because we can handle small amounts, deal with it, excrete it, and we don't have these accumulate. Is there a certain amount of time that you'd have to eat all 18 apples for it to kill you? So I think the way these are actually measured is you're getting that toxic dose in a single sitting. So you'd kind of have to probably take all of those seeds, grind them all up, and then just eat them at once. If you spaced it out over 24 hours, you would be able to handle more than that LD50 amount. So anything that has a CN minus ion group is bad for us. No, it has to be a CN group on a compound that can become free when you digest it. So it's possible to have compounds with CN groups that we eat that our body does not liberate. Yeah, I see a cyanide forms a complex with hemoglobin. I think that's true, because hemoglobin has iron in it, and the iron is like just right to fit an O2 molecule, oxygen. That's its job, is to transport O2 through the body. CO, which is carbon monoxide, is the same size and shape, and it'll fit in there, and it'll block the body's ability to transport oxygen. I think cyanide is the same. Cyanide's the same size, same shape, but it sticks, and it sticks hard. And so the body, it basically knocks out our hemoglobin as it goes. Is there any kind of resistance formed to the body? Would it produce more hemoglobin? Over time, yes. So our body has natural defenses against toxic things that we consume. If you consume small amounts of them over time, the body will respond by increasing the amount of enzymes that are there to detoxify it. And I don't know exactly how that mechanism works. It's pretty fascinating. But for example, if you are a regular drinker, and you drink a lot over a long period of time, your body will respond by increasing the amount of liver alcohol dehydrogenase it produces. So you would develop a resistance to certain things. Now there's of course limits to your ability to do that. Like your body probably would if you had cyanide slowly ingested at like a lower dose over time. I'm guessing the body would probably produce more hemoglobin to account for it. Your body will do that, for example, if you move to like a high altitude where you have lower oxygen in the air, your body will respond by increasing the amount of hemoglobin in your blood. So your body's pretty smart, comes up with mechanisms to deal with these toxins. All right, so despite the fact that these things are quite dangerous, you can actually go on and buy this thing. It's called vitamin B17. It's actually not a real vitamin. If there's any dietitians in the crowd, you can back me up on this. There are B vitamins. They don't go up to 17 though. So what people have done is have taken this compound, this amygdalin, that you find in apples, it's very toxic, and take it from, the pits of other things like the seeds of grapes, pits of peaches, nectarines, whatever. It's in the seeds. They can take it, powder it, and people will sell it as a food supplement. And in particular, people market this as a cure for cancer. This is really terrible because, it's first of all preying on people's desperation. If people develop cancer and have cancer, obviously, people want to survive and they want to try whatever's possible. And so some people may out of desperation just give this a try, when actually it poses a real risk, because what this actually is a toxic substance that produces cyanide in our stomachs and can cause really negative health effects. This is an article from the New England Journal of Medicine, which is a very well-regarded medical journal, where they do a clinical trial looking at the effect of amygdalin, which is the compound that I mentioned that was marketed as an anti-cancer treatment in the actual treatment of human cancer. And what you can see here in yellow, no substantive benefit was observed in terms of cure, improvement, or stabilization of cancer, improvement of symptoms related to cancer, or extension of lifespan. The hazards of amygdalin therapy were evidenced in several patients by symptoms of cyanide toxicity or by blood cyanide levels approaching the lethal range. Amygdalin is a toxic drug that is not effective as a cancer treatment. In fact, I would go as far as to say it's not even a drug. as far as I can tell, there's really no reason to give this in a clinical setting. This is another one. This is a Cochrane review, and I might refer to Cochrane reviews throughout this course a bit. Cochrane review is basically like an organization of people who they don't do direct lab research themselves. Instead, what they do is they create reviews after reading basically hundreds and hundreds of papers on some topic in the literature. Very rarely in the literature does one paper come out, and that's the definitive paper, and that's, problem solved, story solved, we don't have to worry about it anymore. Usually the literature is more like a conversation, and papers get published and they add on each other and they build from each other. Sometimes they correct each other or overturn each other. So what these are people that read all of the papers available on some topic. They rate the papers in terms of sort of quality control and come up with sort of the best summation they can on that particular topic. So this was one that actually produced on the treatment for cancer using amygdalin. The claims that amygdalin have beneficial effects for cancer patients are not supported by sound clinical data. There's considerable risk of serious adverse effects from cyanide poisoning. So this is just another, source, in my opinion, a good objective source, which is sort of casting doubt on this idea that amygdalin can cure cancer. Another journal article here, this was a case study of severe cyanide poisoning from alternative medicine treatment with amygdalin and apricot kernels in a 4-year-old child. So this is the other problem if people start using unproven substances like this as a potential treatment for whatever, is that there is a very strong risk of causing direct harm through poisoning, through acute toxicity, like you see in this particular case of a 4-year-old child. 4-year-old child would have probably a less developed system for handling these compounds, as well as a much lower body mass. So it would take a lower dose to have a toxic effect on someone in that age range. So don't buy vitamin B7, and if you have cancer, talk to your oncologist. FDA is taking action against 14 companies for selling illegal cancer treatments from back in 2017. With the internet, though, these are like what's that thing where you chop one head off and 2 more come back? I don't know. Someone will tell me in the chat. But that's the problem. You can shut down companies selling illegal treatments like this, but then conspiracy theories always arise. People are saying, oh, the government's trying to keep this from you, blah, 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 blah. And it's natural. How can it hurt you? It comes from apple seeds. It's a very, very difficult thing to eradicate, despite action like you see here. The most toxic substance known to humankind. The one with the lowest LD50 of all is botulism toxin. Now, botulism is a microorganism that can grow in food. See, a hydra, not a minotaur. No, those were the things that were like a bull with a human head. Or no, maybe like a bull's head and human body. Hydra. We're getting some more votes for hydra. Botulism is, it's an organism that it's typically found in food that has been canned, but hasn't been properly sterilized. Yeah, minotaur is the bull man. So botulism toxin is produced by this thing, and if you have, typically you find it in things like beans. It's less commonly found in acidic foods or highly sugary foods that are canned. So like jam, it's rare. Tomato sauce is kind of rare. But beans is probably where you most commonly find it. And I would just say, personally, if you're going to eat beans from a can, buy them. Don't try to do it yourself, because you play a huge risk with botulism toxin. You need 2 nanograms per kilogram of body weight if you inject the compound into a vein. That's extremely small. So what that means is 2 nanograms per kilogram. That means if you had a container, like imagine an ice cream container with like 2 liters in it, that container, if that was full of botulism toxin, would be enough to give every human being on Earth a lethal dose. So you don't need a lot of it to cause a really negative side effect. Now, one thing that's interesting though is if you look at the LD50 for inhaling botulism toxin, it's 6 times lower. I mean, the number is 6 times higher, but you need a 6 times bigger dose. So the lethal dose is a smaller amount. So it's 6 times less toxic inhaling versus injecting directly into a vein. There'd be a different number if you swallowed it, and a different number if it absorbed through your skin. So this is another factor that we have to consider when we're thinking about the toxicity of a certain compound. How is it entering your body? There's a number of different ways this can happen. Despite being the most toxic molecule we know of, botulism toxin is also used safely every day by injecting into human beings. Botulism toxin is the active substance in Botox, where botulism toxin is injected not into a vein, but under the skin. And typically you see people get it done, like around their eyes or whatever for reducing the appearance of wrinkles and things like that. So Botox, you can use it safely if you understand the toxicity and the dose and the route of exposure. All of these things can be safe for use in things like Botox, I guess. So there's a number of things that we have to think about when we're thinking about toxicity. All right, next question. Which of the following is not a factor in determining the level of risk that a chemical might pose to you? The route of exposure, the origin, synthetic or natural, the acute toxicity, or the dose? And the correct answer here is B. That's the one that doesn't matter, right? What matters is the molecular structure, the amount, and how it's entering your body. These are the important factors when trying to assess the risk posed by a certain drug or a certain chemical to you. Right. Cool. Good. So now we're going to talk about chronic effects, which is the other side of the coin. Chronic effects are harmful effects that basically are the result of repeated exposure over a longer period of time. So alcohol, if you want to think about the acute toxicity versus chronic, acute, we're talking about LD50, we're talking about poisoning yourself, we're basically talking about overdosing on alcohol. And we know the LD50 for alcohol for human beings is around 7 grams per kilogram of body weight. That if everyone on the stream consumed that much all at once, which we said last time was like 13 shots or something, kind of in one sitting, half of us would die and half of us would survive. If you survive, you might have a pretty bad next day or next 2 days, but 3 weeks down the road, you're probably not going to have really ill effects still from that. However, chronic moderate exposure to alcohol. So the idea here with chronic moderate exposure, what I'm talking about here is like you have, let's say you have 2 beer every night for 40 years. You will never, after that route of exposure, approach your LD50, right? Your body just will get rid of the alcohol faster than it can build up. So you're never going to approach your LD50. You're never really at risk of overdosing on alcohol. However, on the other hand, that will, over time, cause slow, regular, chronic damage. It can increase your risk of developing several different types of cancer. It will risk long-term liver damage, cirrhosis. I always mix up cirrhosis and psoriasis. I think psoriasis is your skin though, and cirrhosis is your liver. I saw a canoe actually, and people like name their boats, and it was called Cirrhosis of the River. which I thought was kind of a cool name for a boat. Anyway, so alcohol will have also these chronic effects. And you can't measure chronic effects the same way you measure acute effects. It doesn't work that way. But can we measure them at all? And the answer is yes, we can. Thanks, Lisa. I miss you. Lisa took the course a few years ago. She started out as a chem major. All right, we're going to come back to the Food Babe here. The Food Babe posted this gem. It's called Chick-fil-A or Chemical Filet. And she complains about the presence of this substance TBHQ in Chick-fil-A sandwiches. This is a compound, actually it's a preservative. It's, psoriasis is the skin. TBHQ is a relative to vitamin E, and vitamin E is an antioxidant which prevents oxidative damage to our cells. TBHQ is also an antioxidant, and it's added to things like fryer oil to prevent those from spoiling, going rancid. It extends their lifetime. So of course, if you fry chicken or fries or whatever in fryer oil, it'll end up in the food that you eat. So she says this: TBHQ is a toxic chemical, it's a chemical made from butane, a very toxic gas, and can only be used at a rate of 0.02% of the total oil in a product. I have the structure there, and I have the structure of butane on the right, and I can guarantee you TBHQ is not made from butane, and even if it was, butane is not a very toxic gas. It's the gas that you find in lighters. And even if it was a very toxic gas, we know that the properties of a substance are not retained if you convert it into some new substance. Just like, sodium is explosive if you put it in water, but sodium chloride is not. So here we go. So we have butane. The reason she says this is because it has this little thing on the side, which is called a tertiary butyl group in organic chemistry. So butyl is in the name there somewhere. It contains a compound. But she says, interestingly, it can be used at a rate of 0.02% of the total oil. Why is there a limit? Maybe because eating only 1 gram of this toxic preservatives has been shown to cause all sorts of issues, from ADHD, asthma, allergies, dermatitis, dizziness, has caused stomach cancer in laboratory animals. So there's a few interesting tidbits here. First of all, she says there is a limit that is allowed by law. And that's an interesting point that we want to come back to, is that the use of these chemicals in public products is regulated. Like the effects of these things have been measured, and there are mandates about the amount that's able to be used. This, by the way, wasn't an original idea from the Food Babe. She took it from this book, The Omnivores Dilemma by Michael Pollan, who says what's quoted there, which is almost exactly the same. He says, but perhaps the most alarming ingredient in Chicken McNugget is TBHQ, an antioxidant derived from petroleum to help preserve freshness. Remember, we don't care where it's derived from, right? We just care about what the structure is. And of course, the structure dictates things like LD50 and whatever else. He says, according to Consumer's Dictionary of Food Additives, TBHQ is a form of butane, i.e. lighter fluid. So they made the same elementary chemical mistake as well in this book. The FDA allows processors to use it sparingly in our food, and then you can see the rest, which was, of course, paraphrased by the Food Babe. He says, ingesting 5 grams of TBHQ can kill. If you go back another one, Consumer's Dictionary, which is where he got it from, you can actually see what the original source was. And this was the definition for tertiary butyl hydroquinone. This antioxidant was put on the market years after pushing by food manufacturers to get it approved. I mean, anything that gets approved gets pushed by their manufacturers, I suppose. It contains petroleum-derived butane, which is not true, and is used either alone or in combination with other antioxidants, butylated hydroxyanisole, hydroxytoluene, BHT. It combines with oxygen very rapidly and becomes brown when exposed to air. That's actually why it's used in fryer oil. It's an antioxidant. It prevents oxidation of the oil prevents it from going rancid, and of course it has the same stuff there. So what I want to point out, though, is they really should have, right from the beginning, hired a chemist like me to, if they were trying to generate fear over this chemical, they missed the boat by saying it looks like butane. Because there's another chemical that it looks much closer to. If you buy anal bleach, which is a substance you get to bleach your anus, the structure of that molecule is hydroquinone, which is actually extremely close to the structure of tertiary butyl hydroquinone. So I think if they marketed this as a close derivative of anal bleach, it would probably have a much stronger effect on the purchasing public, right? So they missed the boat there. Hydroquinone and TBHQ both chemically react in a similar way. Their job is to basically undergo or provide hydrogen atoms to do reduction reactions. Okay, so going back to those levels, according to the Food Babe, the amounts that would have to be present, if you went to Chick-fil-A and decided to eat burgers that were contaminated with this fryer oil that had the maximum allowable amount of TBHQ by law, and you wanted to get the 1 gram dose she said was full of all of those negative side effects, you would end up having to eat 278 burgers to get that level of a dose. Those 278 burgers would contain 5 kilograms of fryer oil absorbed in the meat, and or mostly the breading, and that would contain 1 gram of the TBHQ. You'd have to eat 5 times that amount to get the 5 gram amount that Michael Pollan was saying could cause death. So I'll say this right away. If you decide to sit down and eat 278 Chick-fil-A burgers over, well, certainly one sitting, but even over like a week or a month, you're going to have all kinds of negative biological effects, and it's not going to be the TBHQ that's doing it. It's going to be the fat, salt, sugar, oil, everything else that's in there, or just the sheer amount of food. 278 burgers is probably too many in 1 year of this. This is probably like eat occasionally type of food, not eat 278 days out of 365. So again, you see people bringing up fear-based arguments against certain substances because of, but they miss this really subtle part, which has to do with dose. All right, so let's go forward. A paper came out in 2004. This is by the European Food Safety Authority Journal, EFSA Journal. This is like the Food and Drug Administration, the FDA in the US, except this is the European version of it. And what they do is every so often, they'll take compounds, like food additives that are commonly used, and take a look at the literature, review all the literature that's available that was published since the last time they took a look at it, and update guidance and update recommendations. So this one came out in 2004. Maybe another one is due soon. Maybe they're going to look at it again for all the articles that looked at this safety of this compound since 2004. They did their initial one, or the previous one to this was done in 1987. So what does that work out to? 17 years? So I should look. 2021 would be another 17 years. Maybe we have a fresh one. They said, based on the data reviewed by the panel, they concluded that TBHQ is not carcinogenic and that further genotoxicity studies were unnecessary. They considered the dog as the most sensitive species. So what they did is they looked at basically long-term effects of this compound, chronic effects, not acute poisoning effects, but chronic long-term effects, and they looked at this at different animals. And remember the problem we had before where we said that what you see in one animal may not be directly transferable to what you see in human beings. What they did is they tried it on a couple of different animals, and they chose the animal that they felt was most sensitive to this compound. And it turns out in this case, they thought the dog was the animal that was most sensitive. So what they do is they feed sort of increasing doses of this drug in their diet kind of every day, and I think they do it for a period of 30 days. And they look to see how much they could put in the feed before they start to see any negative effect at all. And I don't mean like kill the animal. We're not talking like poisoning them, LD50. We're just talking about any negative effect. Like, does the animal throw up more frequently than it would otherwise? Is the animal lethargic? Is the animal whatever? Is there a rash? Is there anything that we can identify negative caused by including that chemical in their diet? And it does seem kind of sad to do this with dogs, but that's how they determine things for being safe with people. And what that level is called, it's called the NOAEL, the no observed adverse effect limit. That's the maximum amount you can consume every single day, or what the animal can consume every single day, and still see absolutely zero observable biological effects. Okay, there's no long-term increase in cancer risk, no, whatever chronic effects that you can think of. And once you go higher than that in the animal, then you start to see the onset of these symptoms, these long-term effects. So they do this over a number of different animals. They pick the animal they decide is most sensitive, and it turns out for this compound, TBHQ, the most sensitive animal was the dog, and they said that dogs could safely eat 72 milligrams per kilogram of body weight for the dog every day and not see any adverse effects. So then what they want to do is use that data to come up with guidance for how much human beings can consume. So what they do is they make this assumption that human beings are 100 times more sensitive than the most sensitive test animal. Or really what they're doing is they're building in a factor of 100 safety factor. And when they do this, what they find is if a dog can take 72 milligrams a day per kilogram of body weight, human beings should be able to take 0.7 milligrams per kilogram body weight a day and be within this 100-fold safety factor of still showing no symptoms. So this is way different than an LD50. LD50, we're like killing half of the test population. All we're doing with this is tickling, we're just adding a tiny amount to see if there's any effects at all on their well-being or health. Dividing that value by 100 and then applying that to human beings. So we end up with this number called ADI, which is acceptable daily intake, which is the acceptable amount that human beings should be able to eat and be still within a 100-fold factor of safety from what we've seen in animals. So it turns out if you weigh 100 kilograms for TBHQ, that's 70 milligrams a day, which is your ADI, which corresponds to consuming about 350 grams of oil, which is about a cup and a half of fryer oil from Chick-fil-A, which, I'd argue if you drank a cup and a half of oil from Chick-fil-A every day, the danger to your health is not the TBHQ. The danger to your health is consuming that much concentrated oil, that fat, right? Actually, I got curious one day, and what I got curious about was what is the maximum number of calories somebody could possibly consume in one day? And after a little bit of searching, I ended up on YouTube and found this guy who drank 4 liters of vegetable oil in one day. And basically he just sat down at a table and filled up a glass of oil and drank it. And of course, your body can't process that many calories. Oil is extremely calorie dense. I think it's 9 calories per gram. So, obviously he wasn't able to absorb all those calories, and I think he had a pretty bad next day. But anyway, I don't know why I was talking about that. So here we go. Chronic effects can be measured. First of all, we measure what's called the no observed adverse effect level in an animal species. We extrapolate that to human beings by dividing by what we call a UF, or an uncertainty factor, which is typically a factor of 100. And we turn that into a number which is called ADI, acceptable daily intake. For nutrition, there's a similar thing which is called RDI, recommended daily intake, and that's a different thing. Recommended daily intake is how much you should have at a minimum to maintain health of different chemicals, like vitamins, or protein or sodium or things like that. It's not quite the same thing. ADI is a number you don't want to exceed. Our recommended daily intake for food is the amount you should be aiming to get. By the way, ADI sometimes goes by some different acronyms. I believe some places call it RFD, which stands for reference dose. So if we see reference dose or acceptable daily intake, they really mean the same thing. They're the established safe limit that you can have, and if your food contains higher amounts than that, that's regulated by the FDA. In Canada, it's regulated by Health Canada. So this is around what we're thinking about if you are looking at dose. For TBHQ, the LD50 turns out to be 700 milligrams per kilogram body weight. If you're looking at poisoning yourself acutely and, giving yourself a 50-50 chance of dying or not dying, the LD50 value is, it's a pretty high bar. You need to actually consume quite a lot of a substance to poison yourself with it, typically. The no observed effect level is usually much lower. And it's lower because we're consuming this amount every day, and we're not looking to kill an organism, we're just looking to cause some observable negative effect. Then you take that level, divide by 100, it gets the ADI, and that's the amount that regulators permit places like Chick-fil-A to put in their oil. Assuming, somebody eats a normal amount of some substance, right? Like you're not trying to eat 200 Chick-fil-A burgers in one day. All right, so we have one more question. It says, what is the NOAEL, the no observed adverse effect limit, a measure of? The severity of symptoms a chemical can produce, both chronic and acute toxicity, chronic or acute toxicity. So the answer to this one is chronic toxicity is the NOAEL. The acute toxicity is measured instead by the LD50. So both of those are important numbers to know about if you are looking into exposure that may be repeated exposure of any chemical substance. All right, we do still have some time, so I want to jump to another maybe case study when it comes to toxicity, and maybe another more nuanced form that we haven't really discussed yet, which is the fact that some chemicals actually can exist in slightly different forms, and these different forms can impact us in different ways. And the example I want to focus on today is mercury. So mercury is an element on the periodic table. It is a silvery, shiny, liquid metal. It's the only metal that's a liquid at room temperature, and you can see it in the picture there. It just looks like water, but like metallic water. It's very dense. You may have seen this substance before. It's used like it used to be used a lot anyway in like thermometers and devices that measure air pressure like barometers, though certainly much less. It used to be used, mercury and mercury compounds used to be used in the curing of felt. And felt, if you didn't know, originally was produced from animal furs, animal pelts, and in particular a lot of the European felts that were made like 200-300 years ago used the North American beaver pelts, which of course was important to the early history of Canada and North America. And many people that were involved in using felt, and in particular they were often used in Europe to make hats, felt hats. The people that were making these hats were called hatters, and many of them were exposed to chronic exposures to mercury over many years through working with this substance. And what they developed was chronic neurological damage because of this long-term exposure. They weren't like acutely poisoning themselves. they were getting these chronic long-term effects from exposure to mercury. The neurological damage could manifest itself in all sorts of ways. They may, not have intelligible speech. They may have all sorts of other issues as well. They may have trouble walking in terms of coordination, muscle coordination. And a lot of people just say that they were crazy. And you may know the character, the Mad Hatter from Alice in Wonderland, was no doubt inspired by the fact that hatters very commonly would develop these neurological symptoms over time from long-term exposure to mercury. Now, mercury gets used all the time for different things, and in many cases for health reasons. One thing is, if you have a cavity, what will often happen if you go to the dentist is they'll drill the cavity out and they'll fill it with some substance. And for many years, the substance of choice was called a dental amalgam, or mercury amalgam. What amalgam is basically a mixture of a bunch of different metals together. So they use silver, and they use mercury, and they use small amounts of other things as well. So this is what your tooth might look like. If you have cavities, they'll drill it out, and it looks kind of metallic when it's done. The metal has actually a lot of very good uses, and it's actually like a really safe substance to use to fill cavities. It's durable, it lasts a long time, and has many, many positive effects. Excuse me. Nowadays, what you more frequently probably see instead of mercury amalgams is it's like a plastic kind of substance. It's like a kind of like a gummy plastic substance that they fill it with, and then they use this like UV light to cure it, which makes it hard, and then they can polish it down. So you often see those now. The advantage of those really is that they're white, so you can't actually tell that you have fillings. So what these are actually made of is a roughly 50% mercury, 35% silver, and smaller amounts of tin, copper, zinc, and a few other things. All right, so it turns out if you get exposed to mercury in different ways, it actually matters. If you swallow mercury, only about 0.01% of the mercury you swallow gets absorbed through your GI tract and ends up in your bloodstream. In fact, people actually used to use liquid mercury as a laxative. If they had trouble going to the washroom, they would drink, 50 mils or so of mercury. Just got the question, what would happen if you swallow it? It's great timing. They would swallow it, it would go into their stomach, run through their system, and then, of course, help push things through, come out the other end, and flush, goodbye, problem solved. I don't recommend that, of course, but people would do this and they would be 100% fine. It wouldn't really hurt them at all. And very, very little of it got absorbed on the way through. Environmentally, to me, that sounds like a nightmare because, of course, all that gets flushed and put out into the environment. So that's, to me, maybe the biggest issue associated with that. But, it's actually quite safe. Breathing it in, though, is a different story. Like, you'd be much worse off if you broke a thermometer, for example, in your bedroom, and there was mercury, and it kind of went into the carpet or something, and you just forgot about it. Because that would slowly evaporate, and you would inhale it, absorb 80% of it into your bloodstream, and over time, you may expect to see chronic mercury poisoning effects. So what about use in dental amalgams? Why would we put it into our mouths where we're constantly swallowing food or breathing in if this could cause us problems? So there's a paper that was published here in 2011, Science of the Total Environment, Mercury Exposure and Risks from Dental Amalgam in the U.S. Population Post-2000. And this is now often referred to as the Richardson study after the first author. One thing that I thought was interesting here is Americans have a total of, or an average of 8 cavities per person, where in Canada the average is 4.3. So it's interesting that in the US, for whatever reason, the incidence of cavities is about double what it is in Canada. But really what they did is they looked at the risks, and what they found is that the typical exposure you get from having one filled tooth, one filling, is you are being exposed typically to around 0.5 to 1 micrograms of mercury per day. So if you're the average person with 4.2 fillings, you get somewhere around 3 micrograms exposure per day. The World Health Organization has the daily tolerable intake limit, which is like the ADI for this substance, and they've listed it at 2 micrograms per kilogram of body weight every day. So do the math there, right? You weigh, let's say, 70 kilograms. 70 kilograms times 2 is 140 micrograms per day is sort of the tolerable limit, or the safe upper limit of what you can be exposed to. So if you're getting 3 micrograms per day by having 4 fillings, that doesn't come anywhere close to the 140 micrograms per day that you're allowed. So it's unlikely that this mercury exposure through having filled teeth is going to cause you any issues. He finishes the study with this quote: "All dental devices and materials present some risks. The ultimate goal is to assess the risks posed by all such materials and to weigh those risks relative to one another. It is important not to leap from the frying pan into the fire in an attempt to avoid one defined risk by opting for another material with ill-defined risks." So let's say, for example, you have 4 filled teeth and you got worried about the mercury exposure by just having those in your mouth every single day. And you're like, drill them out, I want them gone, use some other substance. Well, maybe that other substance also leaches something into you by little bits every day, or maybe that other substance isn't as durable and will break down and can lead to a tooth infection, which can lead to other health complications down the road. You can always say what if. So, like he says, trying to avoid some very, very small risk can lead people into jumping into situations that are much more risky. The Canadian Dental Association says this: "Current scientific evidence on the use of dental amalgam supports that it's an effective and safe filling material, provides a long-lasting solution for a broad range of clinical situations. It delivers benefits over other filling materials with respect to ease of use and longevity, especially with patients with a high risk of tooth decay." There's a question: do you know something about the hypothesis or lie that amalgam has some connections to ALS? I don't know about that one in particular, but I've seen dental amalgams erroneously connected to about every possible disease or negative side effect that you can imagine. So while I haven't seen that one in particular, I'm not even a little bit surprised that that's out there. Okay, why is mercury sometimes used in dentistry? The reason there is D. It is used in amalgams to fill cavities. All right. Now I'm going to talk about mercury in a different form. This is dimethylmercury. It's a mercury molecule that contains 2 groups, which we call methyl groups. It's CH3, Hg, CH3. It's one of the strongest neurotoxins known to human beings. This person in the picture, she is a researcher named Dr. Karen Wetterhan, and she actually died due to exposure to dimethylmercury. Apparently she was using it, she was wearing gloves, and she spilled a little bit on her hand, and she knew right away that that was a bad thing. She took the gloves off right away, but in that time, enough had soaked through the glove and then soaked through her skin to give her a toxic dose, which caused her to pass away. The LD50 of this compound is 0.05 milligrams per kilogram body weight, which makes it 20 times more toxic than cyanide. This is incredibly, immeasurably more dangerous, or is a more dangerous form of mercury than that liquid metallic mercury that we saw. This is a substance containing mercury, and the dangers aren't even comparable, right? It's just a totally different world. So this actually connects with the measles vaccine, and actually modern vaccines as well. Measles at one point was an extremely common disease, highly contagious, and these are measles cases in the United States in the numbers of, you can see the hundreds on the scale there, hundreds of thousands of cases, really, is what we're looking at. So if you look at the years from 1944 up to about 1964, the average is somewhere around 500,000 cases in the US per year, and measles can cause all sorts of long-lasting, long-term damage. Cause blindness, it can cause all sorts of things. And you can see these numbers actually, that they're irregular, that they shoot up and they shoot down. And that's because there's outbreaks and then it might calm down a bit, and then there's another outbreak and then it can calm down, and so on. The vaccine for measles came out in 1963, and if you look at what happened around 1963, cases immediately started to plummet. There were a few little outbreaks in the late 60s and the early 70s, and one in the late 80s. As the vaccine kind of got rolled out and it got perfected, and basically it was eradicated. And in fact, I think there was a 5-year span running from, I don't know what years, I think I might have it coming up, where there were 0 cases of measles in the US. They were ready to declare extinct this disease in the US, thanks to vaccinations. Similarly, German measles, you can see a very similar thing. German measles, it's not something you hear about too much anymore, rubella. It was a very serious form of measles, especially to pregnant women. Because what would happen if you developed German measles during pregnancy is it would lead to very serious birth defects. And this is something that actually affected members of my own family, so I'm well aware of what German measles can do. The vaccine, you can see the year it came out was, I believe, late 60s, and since that time the incidence dropped dramatically and was eventually essentially eradicated in the US as well. Now, these vaccines, of course, they're produced by drug companies. They were originally invented not by drug companies, but eventually they took over. Drug companies are exceptionally good at scaling up the production of these things and producing them in a very regulated environment so that they are uniform, meaning every dose looks the same, and safe, and all these sorts of things. Now, originally, what happened, and I believe the way the story goes, is in the early days of, it might have been smallpox vaccine, there was a missionary trip to India to try to vaccinate the population there against, I think it was smallpox, although I could be wrong, it could be something else. And so what they did is they had these bottles, which are called multi-dose bottles. Basically, the bottle contained enough doses for like 25 people. So multi-dose, 25 doses in one bottle. And they flew these over to India, and where they went, they didn't actually have like electricity or refrigeration or anything like that. So when they took these bottles, what they did is on the first day of this clinic, they were immunizing children, and they opened up one of these bottles and drew out, I think it was around 10 or 12 doses, and immunized 10 or 12 kids with this. And then it closed for the day. They took the bottle, screwed the cap back on, put it back on the shelf, not refrigerated or anything like that, and then the next day opened it up again and gave like 15 more doses out of the same bottle. What they found was that all 12 kids that got the dose on the first day were fine. They developed immunity, it worked just like they wanted. All 15 kids that got it on the second day died. And it's extremely tragic, extremely sad. What had happened is when they opened it the first day, it broke the seal, something developed inside of it, it grew overnight in the warm weather, and it caused death in anyone who got an injection out of that bottle the next day. So what they started doing is looking around for something as a preservative to put in the bottle that would prevent bacterial growth or any kind of growth inside the vaccine bottles, inside the vaccine bottles. And what they found is that most common preservatives that they might want to use for this would actually decrease the efficacy of the vaccines until they came across this molecule you see here. Thiomersal is one name for it. Thimerosal is another name. I think thimerosal is a trade name and thiomersal is the name of the drug. But it contains this molecule, and you can see there's a mercury atom in it. This turned out to fix the problem. It preserved it. It didn't decrease the efficacy of the vaccines, and it enjoyed many decades of regular, safe use. And it only really was necessary in what are called multi-dose vials, where you're expecting to kind of open and close the same vial many times. Until 1998, a medical doctor named Dr. Andrew Wakefield published a paper in the medical journal The Lancet, this guy's from the UK, linking mercury in childhood vaccines, in particular the MMR vaccine, which is measles, mumps, and rubella. And he connected it to both autism and inflammatory bowel disease. And he was making this claim that, we should be extremely wary of vaccines. And you may have heard, even as a meme almost, the relationship between vaccines and autism. Well, it all started with this paper in 1998. So it's true that there is mercury in vaccines, or at least in these multi-dose vial vaccines, in there as a preservative. People haven't really studied in great detail the toxic effects of these things. One thing they do know, though, is in the body, this breaks apart here, and what you end up with is Hg with what's called an ethyl group. Sometimes you just go HgEt, the positive charge. However, that molecule does bear a relationship to another form of mercury called methyl mercury. Methyl mercury is found in fish. It's actually when mercury gets into the environment, there's microorganisms in the soil underwater that'll convert mercury in the environment into methyl mercury, and that will work its way up the food chain and can end up in fish. It's present in highest concentrations in fish that are like top predators, things like sharks, swordfish, tuna, and so on. You might have seen an image like this. I know when my wife was pregnant for the first time, we had a table like this that was like a sticker on the fridge that like her doctor gave her. It's trying to regulate your mercury intake. You have to be careful for certain types of fish how frequently you actually consume them. How frequently you might eat them. Particularly if you're pregnant, I guess, because you don't want to cause harm to developing child. So this is a similar form, methyl mercury, and we know this can cause chronic disease in human beings. It can cause a disease called Minamata Disease, which is essentially chronic mercury poisoning. It's called that because of a place in Japan called Minamata. Well, there's a bay called Minamata Bay where there was high levels of mercury contamination, and the local population had a high incidence of this disease from eating fish and shellfish that were fished out of that contaminated site. I have a friend actually who developed Minamata disease from eating tuna fish. She would eat the cheapest tuna fish she could find, and she was eating like 7 cans a week, and that can cause enough of this mercury to build up that you start getting negative symptoms. If you stop eating it, your body will clear it, and then all the symptoms go away. So yes, tuna is one such fish. Albacore tuna, there's different types of tuna, and the different types of tuna can have different amounts. So like you can see, skipjack tuna is on this one, albacore tuna and yellowfin are over here. So yeah, you don't want to eat too much tuna. I think salmon, which is up here, is probably a safer choice because salmon contains much less mercury. So if you're looking for, a fish to eat frequently, that might be one of your best choices. All right, so people kind of assume, well, we know methyl mercury from fish is bad for us. Maybe thiomersal is bad for us too because it has ethyl mercury, which is a cousin of methyl mercury. Not the same, but similar, we could say. To give you an idea, by the way, of the dose of mercury you can expect to get from eating fish, if you get half a can of tuna, so if you make, you open a can of tuna, you can make roughly 2 sandwiches out of that. That contains about 75 micrograms of mercury in the form of methyl mercury. The World Health Organization, tolerable daily intake, which is basically the same thing as acceptable daily intake, 3.2 micrograms per kilogram of body weight. If you weigh 70 kilograms, what does that work out to? Like 220 micrograms per day if you are like 70 kilograms. And so you can have your tuna fish and probably stay under there pretty easily. If you had a whole can, you're going to get double that, and depending on your body weight, you can see how, you don't probably want to eat a can of tuna fish every single day. One flu shot that is stabilized with thiomersal will have 25 micrograms of mercury in the form of ethyl mercury. So you get about 1/3 of the dose from getting a vaccine of mercury than you would from the sandwich, from eating a sandwich. So, again, like you could safely eat a tuna fish sandwich now and again. Don't eat 10 of them a day, right? Same thing with flu shot. You can get a flu shot and you can be very, very safely dosed with ethyl mercury, but don't get like 25 flu shots every day, right? Like that's overwhelming. So what happened is when this paper came out in 1998, they decided as a precaution, just let's remove it from all childhood vaccines. And the way we can do that is get rid of these multi-dose vials. Get rid of these bottles that have 15 or 25 shots. We'll have what's called single-dose vials. So every vial contains 1 shot, and then it gets chucked. That way we don't have to worry about opening it and closing it and opening it and closing it. We don't have to worry about contamination, and therefore it's a one and done. This was the solution in 1999. Let's just get rid of this compound and then study it and see, is this connection that was published in 1998 by Wakefield really the case? So out it comes. Turns out, after much study, it turns out ethyl mercury is much less damaging to the body than methyl mercury, and what it really comes down to is the body's ability to clear it. Mercury can build up in your blood over days, months, and it actually has a half-life in your body of about 50 days. So what that means is if you ate a tuna fish sandwich with 75 micrograms of mercury, 50 days later, half of that mercury is still circulating around in your body, assuming you absorbed 100% of it. Ethyl mercury, the body is actually a little bit more efficient at filtering it out of your blood and excreting it, so it has a half-life of only 18 days, so it doesn't build up as much, it doesn't hang around as long. After 50 days, or after 100 days, only 2% remains in your blood, whereas the methyl mercury from the sandwich, roughly 1/4 of it is still there after 100 days. So ethyl mercury turned out to be much safer than the methyl mercury that we eat when we eat certain types of fish. But then even worse, not only did it turn out to not really be as bad as was originally suspected, even given the fact that you get a small dose and extremely infrequently, many other organizations took up the claims and investigated them and did additional study. And the following came out and said, we can't find this link that Wakefield proposed in 98. CDC in the US, American Academy of Pediatrics, Institute of Medicine, US National Academy of Sciences, Cochrane Library, which we mentioned before, UK National Health Service looked at it as well. Basically like, we're studying this and where are you getting your information? We're not seeing the same thing. What's going on here? And they did an investigation on Wakefield's paper and they found out a bunch of very troubling details. First thing was that Wakefield was paid £55,000 by lawyers of a group of parents that were suing the MMR vaccine manufacturers, claiming the vaccine caused their kids autism. Problem is there was no evidence that MMR vaccine was linked to autism, so essentially they paid Wakefield to produce a paper to support their legal claim. Wakefield also at the same time had applied for a patent for his own vaccine that would compete with the MMR. So what better way to pave the way for him to sell lots of his own version of the vaccine than to have the current one on the market banned? During his research, it was found that he performed invasive procedures on 12 autistic children, such as colonoscopies and spinal taps, without ethical approval and contrary to their clinical interests, which means he didn't do it because they needed it for some medical reason. The next one gets me. He had a birthday party for his son and paid the kids that came to the party £5 each to let him draw blood samples from them, and he used the blood samples. I mean, that's not how we do research. Furthermore, they looked at his results that were published in his paper and found out that he just made up numbers, that the numbers that he was publishing weren't the same ones that were in the lab books that were kept when the research was done. Chimodia asks, how does one more carbon atom make such a significant difference? It's amazing how one atom can often make a huge difference in how something can affect us. The example I like to think about is vancomycin, which is an antibacterial drug. There's a bacteria that has developed resistance to it called vancomycin-resistant Staphylococcus aureus, and it developed resistance to vancomycin by changing one atom in peptide chains that hang off the membrane. It's amazing, and there's lots of different reasons why one atom can cause such a big difference. Different mechanisms, maybe I should say. Morgandy asks, why didn't they take away his doctor's license? Well, you just wait. It's published as fraudulent. This became big news, of course, and the British Medical Council charged him with 36 charges, 4 charges of dishonesty, 12 charges involving the abuse of developmentally challenged children. The Lancet in 2010 went back and retracted the 1998 paper and stated that elements of the paper were falsified. And in May 2010, he was barred from practicing medicine in the UK. He's not licensed anywhere else now either. So they did take away his license, and really, like, professionally, it kind of basically destroyed him. Although he's sort of become, in the anti-vax community, a bit of like a martyr, if you know what I mean. It's like, oh no, people will say, like, he was just speaking the truth and the corrupt system shut him down and all this sort of thing. So he certainly hasn't retracted himself. Most of his co-authors have, but he himself sort of remained a staunch supporter of his original findings. So this is what the MMR vaccine coverage in England looks like. It was in the low 90s every year up to around 1998, which is when his paper came out, and then it started to dip, and it dropped down to about 79%, and then once the fraud became uncovered, 2010 was when his medical degree stripped from him. You know, also what happened in the early 2000s was a super heavy push by National Institute of Health and Health Canada and so on to promote MMR as a safe vaccine, realizing that in 1998 they took the mercury out. There's all kinds of people that still believe this. But what you see, since that time, vaccination rates have recovered. They're kind of back up to where they were before this. But in between here, there's this big gap. There's this generation of people with inadequate vaccination status. It turns out that vaccines impart something called herd immunity, and that if you get to a certain percentage of the population that's vaccinated, herd immunity will prevent spread, prevent outbreaks. The herd immunity percentage you need to hit is different for different diseases. It depends on how easily those diseases get spread. But we see this temporary dip in vaccine coverage, and this has led since to various measles outbreaks. This was one in Washington State. This is a few years ago now, but there was 36 confirmed cases, 12 suspected cases. Here's a really bad one that happened in the Democratic Republic of Congo. Over 6,000 people were killed. And where this was found to originate, this first outbreak a couple of years ago in the US was actually Disney, and it was among these clusters of unvaccinated people that really this anti-vax movement kind of began as a result of. So again, the risk from mercury in vaccines is way lower than the risk of the disease you are exposing yourself to or making yourself exposed to by avoiding the vaccines altogether. All right. So we're at 2:27. I think maybe what I'll do, how many slides do I have left? I'm going to talk about this next piece, which is around another chemical that you hear about a lot, maybe, especially if you like to frequent alternative news sites and things like that. Glyphosate is a chemical that gets a really bad rap. It's marketed as Roundup. It used to be Monsanto, but Monsanto got purchased by Bayer, I think. So it's a Bayer compound now. It has this structure. It is an organic molecule that if you look at that part of the molecule, that part I circled is glycine. Glycine is an amino acid. It's found in almost every protein. And it has this phosphate group that's attached to it. So it's glyphosate. Gly for glycine and phosate because it has that phosphate group, the phosphorus in it on the other side. This is used as a pesticide, particular an herbicide, which means it's there to kill plants. It's an extremely effective. Basically, it's effective against all types. If you look at its toxicity, it actually has a really, really low acute toxicity. It has an LD50 of 5,600 milligrams per kilograms of body weight in rats. That means in one sitting you would have to drink 27 bottles of this to hit your LD50 of this substance. There are surfactants, which are basically soaps that are also present in this bottle, and the soaps themselves have a higher toxicity than the actual active ingredient, the glyphosate. And this is based on testing on rats. So yeah, unless you drink 27 bottles of this, very, very, very low risk of poisoning yourself. That's not true for many different types of herbicides and pesticides that are used in agriculture. So this actually was widely embraced by agriculture because it has such a great toxicity profile that farmers could use it and have a very low risk of exposing themselves or their animals or their children or whatever, or their neighbors, to toxic levels of this substance. Yeah, Bayer is the new owner of Roundup and many seeds throughout the year that have been sterilized. Chronic effects for rats is 31 milligrams per kilogram body weight. That means if rats consume less than 31 milligrams per kilogram body weight every day, they see no effects. That gets converted to an ADI for humans by dividing by 100. So what that means is you can consume 22 milligrams of glyphosate per day and remain within the 100-fold margin of safety of the NOAE level established in rats. So you see papers like this: widely used herbicide linked to cancer. And then you see all these other agencies like European Food Safety Authority, Health Canada, European Chemicals Agency, United States Environmental Protection Agency. All of these say glyphosate is unlikely to cause cancer in human beings. World Health Organization does come out and say it is linked to certain types of cancer. They call it a probable carcinogen. So why is it that this one agency is saying that glyphosate, which has been established, has established safety limits and so on, does cause cancer, whereas these other ones do not? An interesting one is that the World Health Organization, International Agency for Research in Cancer, is one branch of the WHO, whereas we have the Food and this is also a branch of a similar organization. Two different branches of the same place, giving different impacts. What we find out is they're actually both correct. but they're speaking different languages. And I don't mean like one's German and one's English. I mean like when they say it's unlikely to cause cancer in humans, that's different from saying it can cause cancer in humans. And this comes down to what the difference is between what we call risk and what we call hazard. So I'm going to explain it like this. A hazard is something that has the potential to have chronic biological effects at some hypothetical level of exposure. Risk is the likelihood of it actually having those effects on human beings. And that might seem like slight variations of the same definition, but I want to illustrate it like this. If you live in the valley or have lived in the valley for very long, you may have done the Cape Split Trail. And I realize this map is now out of date because there's a new loop. I think it was just new last spring. So I think I don't know what it does. It comes back here and then it reconnects at some point. Maybe it reconnects all the way over here. I don't know. Anyway, if you've done Cape Split and you made it all the way to the end, you end up basically right here. Very, very beautiful view. You can look out across, but you see these huge cliffs, right? I don't know what they are. They're at least 100 feet high. If you fell off that, you'd definitely fall on the rocks below and probably die, right? Almost certainly you'd die. So what I'm saying is that that cliff presents a hazard. It's a hazard in that it could kill you if you fall off of it. But if you do this hike, what is your risk of falling off the edge? Well, if you do like me and maybe come down to maybe around here and stop, sit down on the grass, enjoy the view for a minute, turn around and walk back, the risk is extremely low. There's almost no worry that a gust is going to pick you up and blow you off the edge. But imagine instead you get on a unicycle and like drive around the perimeter, ride around the perimeter as close to the edge as you can on your unicycle. There, your risk is extremely high. In both cases, the hazard, which is the cliff, remains the same. The hazard does not change. The hazard's the same, it's the cliff, no matter what. What changes the risk is your behavior. So this is when we come back to a chemical. A chemical may pose a hazard that you can completely eliminate by making decisions that cause your personal risk to be very small. And so when we look at something like glyphosate, it is possible, for example, if you drank 10 gallons of glyphosate every week, that over long term, that could increase your risk of developing certain cancers. That does not mean that if you read the instructions, follow them safely, do what you normally do, have a normal diet, etc., that you are putting yourself at any risk at all. This is why different agencies come out with these different rankings. By the way, this actually happens. Some people do hurt themselves. This was a story from 2016 where a hiker was seriously injured falling trying to save the dogs that went over the edge. So people certainly do hurt themselves here, fall over the edge. But a lot of people do this hike with no danger or risk to themselves at all. So what the WHO does is it has these classification categories where they're classified in terms of carcinogenicity. And it's not in terms of like how likely they are to cause cancer to you, it's how well we know whether they cause cancer or not. And it's nice to know there's only one single chemical that is listed as probably not carcinogenic. And that chemical is caprolactam, which is a chemical found in nylon. That's the only substance ever studied that has been definitively shown, or not even definitively, probably does not cause cancer. Group 1 means we know very well that these substances cause cancer, and the data is very strong. So what this scale is the strength of our confidence in the data, as opposed to potency of the substance in terms of causing cancer. You see things in this list, we have things like tobacco, we have plutonium, processed meats like bacon, alcohol, sun. If you get too much sun, you can develop skin cancer, etc., etc. These are all things that we know and well understand that they cause cancer. Probably carcinogenic to humans. has a lot of interesting things. It's not just substances, but things like working as a hairdresser probably increases your risk because of various occupational exposures. Red meat probably increases your risk of developing certain cancers. Very hot beverages, if you drink your tea too hot, that can increase your risk probably of certain kinds of cancers. There's all kinds of things. Working night shifts, if you work shifts like a nurse or something like that. Getting less than 6 hours of sleep is also in this list. And glyphosate is in that list. That does not mean that you've eaten vegetables that were grown in a field where glyphosate was sprayed. That does not mean that type of exposure is going to cause you cancer. It just means there is a hypothetical exposure to this substance that probably would cause cancer. Okay, that's the difference between hazard and risk. This thing in this group 2B are things that are like probably, possibly carcinogenic. We just don't really know. And then carcinogenity not classifiable. We don't really have enough good data to say one way or another. Is there anything at all? And this is 505. Like that's the majority of the compounds studied. All right, so I think we're coming around to the end of this unit. We're ready to conclude soon. First one is all substances can have acute effects and chronic effects, including things like water. Magnitude of these effects on us depends on dose, route of exposure, other effects. Even if you think about drugs, other drugs you're taking at the same time could impact that as well. 2, acute and chronic effects are knowable. They're measurable. We measure them in animals typically and extrapolate them to human beings. Use of any chemical involves some level of risk and potentially some benefits as well. We wouldn't be using substances if they didn't have benefits. National and international agencies like Health Canada generally very trustworthy and largely agree with each other on the dangers posed by certain things and safe ways of handling them and so on. Detox, diets, teas, foot wraps, soaps, candles don't do anything to reduce toxic load in your body. They may give you a peaceful state of mind, and if so, fantastic, but they're not going to extract toxic substances out of your body. They don't work. We have one more question. Which discredited medical doctor played a large role in fueling the anti-vax movement by his fraudulent paper linking vaccines and autism? The answer here is A, Dr. Andrew Wakefield. We're going to talk about some of the others later. Well, we talked about the food babe a couple of times. Dr. Oz, we'll talk about him a little bit too. Dr. Phil, I don't know if he comes up or not. All right, so that's really where we're at right now. We are at the end of today's lecture. We're at the end of this unit. What that means is the quiz for today's unit will remain up for 1 week starting today. So the due date for that will be next Tuesday. And what we're going to be doing is starting our next unit this coming Thursday. So I'd like to say thank you to everybody, and I will see you all on Thursday. If you need anything from me, if you want to talk to me about anything, please send me emails. If you have stories or personal experiences or anything like that that are kind of linked to what we're talking about in class, I'm always happy to hear them. I'm always happy to chat with you and hear any insight you might have. So please let me know. If you have questions, send them to me too. I'm kind of available anytime. All right, we'll see you all later and have a great couple of days.